All night aspirants, you are listening to podcast Botany for NEET by S. Ramakrishna. In this, the topic molecular basis of inheritance and the subtopic RNA world. RNA genetic material, initial genetic material, it was RNA. What are the evidences to support that RNA was the initial genetic material? Because life began around RNA. RNA, it was a central. The total life process like translation, metabolism, splicing evolved around the RNA. If we take the translation, ribosome help in peptide bond formation. Ribosome is RNA nucleotide. And splicing, it involves snRNA, small nuclear ribonucleic acid, along with the protein involved in the splicing of heterogeneous RNA. And metabolism, ATP, NADP, NAD, which are the nucleotides. So there is involvement of RNA. And RNA genetic in few viruses, QB bacteriophage, tobacco mosaic virus, and HIV. And RNA performs some reactions which are catalyzed by the nucleic acid enzyme, that is ribosome. So certain reactions are not catalyzed by the proteinaceous enzyme, but a nucleic acid enzyme, that is ribosome. And RNA, as it is catalytic, why it is catalytic? Because it is chemically highly reactive. Why chemically it is highly reactive? Because of the having 2 OH at every nucleotide. So RNA prefers quick degradation as well as quick synthesis. So later protein took a role of complete catalyst. As RNA is labile and degradable, DNA evolved from RNA by undergoing structural and chemical modification. What are chemical modification? DNA acquired thymine in place of uracil that provides stability. That is one thing. And structurally, it became double-stranded molecule. So DNA resists the change because it adopts a process of the repair. Both RNA and DNA undergo mutation, but uncontrolled mutation prevented in case of DNA. This balance makes the DNA it is highly stable. And any mismatched nitrogen bases are integrated with a proofreading and repair mechanism. It is going to be checked by repair mechanism. So DNA it is highly stable. Next, DNA replication. Watson and Crick proposed double helical model of DNA and also proposed replication of DNA. To quote their statement, original statement of Watson and Crick, it has not escaped from our view the specific base pairing we postulated also suggested a copying mechanism of the genetic material. This is a great revolutionary step in case of the DNA. Why it is a revolutionary step? Simplicity in explaining genetic implication with regard to the structure of DNA as a genetic material. Because with replication, two daughter DNA are formed identical to parent DNA. Why two daughter DNA identical to parent DNA? The two strands of DNA act as template. Each strand synthesizes a complementary strand. That's why two daughter DNA molecules are formed identical to parent DNA. So it obeys the general rule of heredity or inheritance because progeny is similar to parents. Here two daughter DNA similar to parent DNA. And what do you call this mechanism? It is semi-conservative mechanism. In every daughter DNA molecule, one parental strand, one new strand present. That's why it is semi-conservative mechanism of DNA replication. But this semi-conservative replication of DNA, no experimental proof by Watson and Crick. It was fulfilled by Michelson and Stahl, first in E. coli, later some other scientists in case of the plants as well as animal. So first semi-conservative replication experimentally proved by Michelson and Stahl in E. coli, a prokaryote having circular, naked, double-stranded DNA, and who employed a method autoradiography and a cesium chloride centrifugation. Cesium chloride is not meant to identify DNA based on complementary bases. It distinguished DNA based on densities. The DNA with high densities and low densities, the DNA sediment at different position based on its densities. The heavier DNA prone to gravitational force present at bottom of the test tube, it sink. Light it become beyond, and hybrid it is semi beyond. So sink, semi beyond, beyond. So N14, N14 it is a normal nitrogen, it is beyond. N14, N15 DNA it is semi beyond. N15, N15 it sink at the bottom. So light DNA, hybrid DNA, heavy DNA. Light DNA both strands are with N14, N14. Hybrid DNA N14, N15. And heavy DNA N15, N15. Next, the DNA of E. coli bacteria labeled with N15. So both strands are labeled with N15. N15 also incorporated into nitrogenous containing compound. And in case of zero time, after centrifugation, the heavy DNA present at the bottom, sediment at the bottom. So the single band, one DNA molecule, N15, N15. Next, this is allowed to replicate in case of the medium having deoxynucleotide with a normal nitrogen, N14. In first generation, it is 20 minutes, second generation 40, third generation 60, fourth generation it is 80. In first generation, it is allowed to replicate in N14. Two strands will separate N15, N15. An opposite strand N14 formed. N14 formed. So the two DNA are hybrid. It is one DNA with N15, N14, other also with N15, N14. Two DNA molecule. So it is semi-beyond, present in case of middle of the test tube. There is no N15 DNA molecule. In next 40 minutes, that is second generation, the strands of DNA will separate, N15, N14 will separate. So 4 DNA molecules are formed. Last 2 DNAs, which are N15, N14, 2 hybrid, 2 light. And in case of third generation, that is first generation 20 minutes, second generation 40 minutes, third generation it is 60 minutes. 4 DNA molecule become 8 DNA molecule. In 8 DNA molecules, the number of light DNA molecules number will increase. That is, the 6 DNA molecules are with N14, N15, and remaining 2 DNA molecules with N15, N14. So out of 8 DNA molecule, 6 DNAs are N14, N14 strand, and remaining 2 DNA molecules each with N15, N14. So total it is 8. In case of the 80 minutes, first, second, third, fourth generation, the 16 DNA molecules form. In 16 DNA molecule, 14 DNA molecules are N14, N14, remaining 2 N15, N14. So what it suggests, number of light DNA molecule increases mean it shows semi-conservative replication of DNA. So cesium chloride centrifugation helps us to distinguish hybrid, light and heavy DNAs. Next, suppose hypothetically the question it is like this. If DNA of E. coli incorporated with N14 allowed to replicate in case of N15, how many DNA molecules are formed with N15 after 80 minutes, the fourth generation? So 16 DNA molecule, the 14 DNA molecule are with N15, N15 because it allowed to replicate in case of N15 medium, and 2 DNA molecules are with N15, N14. So If it is incorporated with N15, allowed to replicate in N14, that is one thing. Incorporated with the N14, allowed to replicate in case of N15. So first generation 20, second generation 40, third generation 60, fourth generation it is 80. Next, the semi-conservative replication of DNA experimentally proved in case of eukaryote by Taylor and associate by using radioactive thymidine in Vicia faba, that is faba bean that belongs to Fabaceae. That means in chromosome also DNA replicated in a semi-conservative mechanism. One chromatid, one chromosome means to one DNA molecule, two strands are present. If you label with the radioactive thymidine, two strands in case of one chromosome, both are labeled. In next generation, the two chromatids are found, each chromatid with one parent DNA, that is strand, other with what it is the normal strand. So, likewise, in case of E. coli, in this also the DNA replicated in semi-conservative mechanism. So, prokaryote first by Michelson and Stahl, eukaryote by Taylor et al. So, in case of E. coli, circular DNA, Vicia faba, linear DNA. In this machinery of enzymes in DNA replication, even though different enzymes are not mentioned, for better understanding of DNA replication, the concept, you must have the peripheral idea with regard to the enzyme. The enzymes involved are helicases, DNA polymerases, DNA ligases, RNA primases, topoisomerases. In these enzymes, DNA polymerase is very important enzyme. The word itself conveying its meaning. It polymerases deoxynucleotide. Thus, DNA polymerase is highly an efficient enzyme. Why it is an efficient enzyme? What factors attribute to call it as an efficient enzyme? Because it works with a high degree of accuracy. It holds deoxynucleotides only but not ribonucleotide. So, if any sort of mistake, that leads to mutation. It does proofreading correctly, but any sort of mistake leads to mutation. Second, not only with accuracy, with a high speed, that means polymerization has to be very fast. If you take E. coli, it is having haploid content 4.6 into 10 to the power of 6 base pair, in contrast to diploid content of the human 6.6 10 to the power of 9 base pair. In E. coli, for 4.6 into 10 to the power of 6 base pair, it took 18 minutes at the rate of 2000 base pair per second. Approximately it is 18 minutes. Next, for this polymerization, deoxynucleoside triphosphate act as a dual role, because deoxynucleoside triphosphate or deoxynucleotide diphosphate, it acts as a substrate for polymerization, and 10-2-phosphate provide energy for polymerization like it is ATP. Next, as helicase unwinds the DNA by breaking the hydrogen bonds, two strands unwinded. So, unwinding causes some pressure above replication fork. That pressure is reduced by topoisomerases. Why unwinding occurs at particular some places? Because two strands cannot separated at a stretch in one go. So replication occurs in a small opening, what we call replication fork. So as helicase unwind the DNA, two replication forks are formed. So one replication bubble, two replication forks. And the DNA polymerase only in case of 5'3" direction. It is unidirectional. But DNA strands which are anti-parallel, that creates additional complexities at replication fork. As it is anti-parallel, but DNA polymerase only in 5'3" direction. On one strand exposed with 3OH, other strand exposed with 5-phosphate. So on one strand very smooth, there is a continuous synthesis of the DNA. What do you call it is leading strand. So leading strand synthesized in 5'3" direction and it is towards replication fork. So leading strand formed on a strand with a polarity. That means leading strand template polarity is 3,5. On other strand exposed with the 5-phosphate, for polymerization is not continuous, it requires the help of the primers. It acts as an anchor that provide 3OH. So this acts as a primer. It is a ribonucleotide. RNA primase involved in the synthesis of RNA, a short oligonucleotide. By obeying the rule of complementarity, it acts as an anchor. It provides 3OH. So new deoxynucleotides are added at 3OH. So DNA formed in fragments on strand with polarity 5'3". These fragments, what do you call Okazaki fragment? Later these fragments are joined by DNA ligase, later RNA primers which are removed. So whatever the discontinuously synthesized strand later joined by DNA ligase, that strand what do you call lagging strand. So lagging strand formed on a strand with the polarity 5'3". So overall leading strand synthesized in 5'3" direction, lagging strand synthesized in 5'3" direction, leading strand template is 3,5, lagging strand template is 5'3". DNA polymerase reads both leading as well as lagging strand in 3,5 direction only because it synthesizes in 5'3" direction. Next. So leading strand towards replication fork, lagging strand away from replication fork. So overall it is one strand, it is continuous, the other it is discontinuous, overall it is semi-discontinuous. So replication is semi-conservative, semi-discontinuous, bidirectional. From replication fork it moves both forward as well as what it is the downward direction, backward. That's why it is bidirectional at a replication fork. So semi-conservative, semi-discontinuous, bidirectional. Next. Not only DNA polymerase is the enzyme for replication, other enzymes also works for high degree of accuracy. And DNA replication not initiated randomly but initiated a particular place, what do you call it is ORI, origin of replication. If you take prokaryote, it is having one origin of replication. If you take eukaryote chromosome, many origin of replication. So origin of replication where DNA replication is initiated. That's why in recombinant DNA technology, if you want to propagate a segment of DNA to multiple copies, it needs a vector. Vector provides what? ORI. That is one point. And DNA replication not followed by cell division. Suppose one chromatid, it is accommodated in one DNA molecule. After replication, two chromatid, one chromosome. The two chromatid, one chromatid distribute to one nucleus, other chromatid distribute to other nucleus in normal condition. Replication of DNA not followed by division of the nucleus results in endomitosis or endoploidy. 2n, 4n, 8n, the ploidy will increase. What do you call endomitosis or endoploidy? It is an abnormal situation seen in case of cancer cell. So replication of DNA not followed by cell division, that is karyokinesis, results in endomitosis or endoploidy. In this it is the replication. In this the key point. This replication in case of the prokaryote, eukaryote, E. coli, and in case of eukaryote, that is chromosomes. It is having one ori, it is having several oris. Which is the enzyme helpful for DNA polymerization? It is DNA polymerase. The enzyme which unwind the DNA during replication, helicase. You have to connect this to transcription. In transcription also DNA unwinded, but it is by RNA polymerase. In this also unwinded, but it is helicase. So helicase involved in unwinding of DNA during replication of DNA. RNA polymerase involved in unwinding of DNA during transcription. Next, which is the point in case of vector which is responsible for multiplication of the alien piece of DNA to multiple copies, ORI, origin of replication. And this replication is semi-conservative, semi-discontinuous, bidirectional. As DNA do not separate at a stretch in one go. Why? It is a high energy expensive process. That's why replication occurs in small opening called replication fork. How many replication forks are formed? 2. How many replication bubble? It is 1. How many origin of replication? It is 1 in case of prokaryote. Leading strands incisor in 5-3 direction. Lagging strands incisor in 5-3 direction. Leading strand template is 3-5. Lagging strand template is 5-3. Leading strand formed towards replication fork. Lagging strand away from replication fork. And DNA polymerase reads template in 3-5 direction because it's incised in 5-3 direction. I am signing off from this episode. In next episode, you'll see in case of the transcription and genetic code. If you feel this approach is right, don't forget to subscribe and share to the person who is seriously preparing for NEET.